Happy Thursday morning. I believe that we are ready to make a start with our morning session and have a bit of a look of two past chapters. I know that we had a look through the chapter on urinary system last week. And it turns out that that was actually a rather quick week. There was a lot going on that week. So I want to take a moment just to make sure that we have taken the time to really discuss the key points of that in case anyone wasn't able to attend the face-to-face class where we had a quick summary of that. After that, we'll do also kind of a reminder of the material that we have covered today or this week on the fluid balance. So these two chapters go very nicely together. So that's the plan for the day, and it's really aimed to be more of a crash course or a recap of the things that we've covered. For the full story, I would refer you to go back to the full lecture videos, which I have made available for you on your Canvas course shell. I think this little picture is a good place for us to start, though. You know, I'm very big on highlighting that things should come together and we should understand the bigger picture. Although our course has been structured on systems approach, where we look at one system at the time, moving on to the next system and next system and next system, it is important to appreciate how all the systems in the body work together. And what we're seeing here is really, I would even go as far as saying pretty much most of that your body ever does. It combines multiple systems, shows multiple physiological processes and their end goals, their requirements, and what we are left with to deal as a result of trying to reach those goals. So we have talked earlier on about the respiratory system and how one of the main goals of the respiratory system was to bring in oxygen. The reason why we brought in oxygen was that the cellular respiration could take place. So in cellular respiration, we convert sugars to ATP. To do this job, we of course needed the oxygen. Well, cellular respiration is one of the basic concepts that I would expect that you have covered back in your introductory biology classes. It really is what's needed for all cells to do their functions. We do need to produce ATP, which was there, if you remember, I defined it as a cellular energy currency. So what drives most reactions, most processes in the cell? Now, if we have a look of the waste products that result from this cellular respiration, what we'll notice is that we are left with carbon dioxide and we're left with water. So we need to get rid of this carbon dioxide and water somehow. And of course, here comes the challenge of transporting them. And to transport them, we recruited our cardiovascular system. And what we saw that we could transport carbon dioxide and water by converting them into carbonic acid. Or we could convert carbonic acid further into bicarbonate. And it is really as bicarbonate that most of the carbon dioxide is transported in the blood, and then it travels to the lungs, and we can then breathe the carbon dioxide out. So far we have connected our, if we want to add here, our digestive system, which we use to get those sugars into the body. We have also talked about the respiratory system. We have talked about the blood, and blood as an special part of the cardiovascular system for transporting these gases, we got back to the respiratory system. So we have tied in multiple systems together. One thing that we have not addressed so far from this process of cellular respiration and resulting water molecules is that what are we going to do with these hydrogen ions that we are left with from the carbonic acid equation, converting carbon dioxide and water into carbonic acid, then to bicarb. As we converted these things to bicarbonate, we are left with excess ions, and we need to do something with these hydrogen ions. And what we do actually get rid of them largely through the urinary system. So that's the system that we recruit to assist with that. So you can see how really it is hopefully making sense while we're covering all of these systems connected to each other. It is pretty much impossible to appreciate the whole body view, the global view of the body without seeing these connections between these systems. So although for this last week's chapter and this week's chapter, our focus is on the urinary system, it's important to know that without urinary system, we would really run into issues with this buildup of excess hydrogen. We would run into issues of not being able to sustain other processes that are taking place. So let's have a look of the urinary system and try to get a little bit more sense of that. What I'm expecting is that a lot of the urinary system is familiar to you from the past, and today we're simply taking that knowledge and adding a little bit more detail to the level that we're looking at the system now. So in the urinary system, there is going to be 4 specific parts that I assume that you are quite largely familiar from the past, but I do want to remind you of those. So let's just jump right to it. So we will have 2 kidneys, and we'll discuss about those, and those are really the business end of this system. So we'll talk about how kidneys are the major organ, how their functions have to do largely with filtering blood. And through this filtering blood, we're then able to regulate fluid volume, concentration, blood pressure, red blood cells, vitamin D, all of those side aspects of the story. And as we saw, we had 2 kidneys. From each kidney, we have a ureter that runs out and transports the urine into the urinary bladder. So we'll discuss a little bit about ureters, what's specific about their characteristics, and what do we need to know from those. Then we get to the urinary bladder. Now, unlike kidneys, which we had 2, and we had 2 ureters, there's going to be only 1 urinary bladder. And urinary bladder's role is really to work largely just as a temporary storage reservoir for the urine that we produced. So we don't have to go to the bathroom all the time. We can build up the need to go to the bathroom and utilize that when it is appropriate time and we have appropriate place for that. And finally, the last structure that I want to discuss is the path out from the bladder to the external world, which is going to be our urethra. And one thing that we'll notice, there are going to be large differences between male and female urethra. So I think that about summarizes the key structures that we'll be discussing. When we get to the kidneys, there is something more I want to highlight in terms of the structural and functional key unit of the kidney, and that is going to be our nephrons. So we'll spend some time talking about those as well. So if you are not familiar with the nephron from your previous studies, this is the time when it definitely becomes important for us to get familiar with these microscopic but extremely, extremely important structures. So if we talk about the kidneys, really the biggest take-home message that I would probably give out of those is their main function in filtering the blood. And through filtering blood, then we're able to do a lot of other things, such as regulate the total fluid concentration, but also volume, regulate concentration of extracellular, intracellular fluids, acid-base balance. We'll discuss about that in the next section, which was this week's material. Of course, we're removing a lot of materials with the urine as well, so waste products, toxins, drugs, and so on. Kidneys do play important role in the in our hormonal system, especially when we look at the erythropoietin and renin, two important hormones for our endocrine system. So we're kind of getting back to where the entire course started. I hope that that kind of reminds of these connections again. And of course, kidneys, we have many other processes that are worthwhile to note with them as well. If we look at the external anatomy of the kidneys are these reddish brown. The color depends a little bit whether you're looking one in an actual patient or if you're looking one that has been dissected, one that has been embalmed. The color may change a little bit, but they are these brownish colored structures about, in terms of if you look at the shape, kind of resemble us a little bit of the bean. This shape is going to be important when we are looking at different parts within the kidney. What we'll notice is that the medial surface is going to be concaved, as you see here on this little dotted line that I've added there, just to emphasize that. Whereas the lateral side is then going to be our convex surface. What I want to show and what I want us to focus at this point is to the renal hilum, and renal hilum as it relates to this concaved medial surface. So what we'll see is that this is a really busy part in the terms of the external anatomy of the kidney. This is where a lot of vessels are coming in. So our renal artery, our renal vein, also in addition our urethra, our ureter. So let's get that here. And we also have, in addition, some lymphatic vessels, nerve supply, and so on. If we look at the kidney, just the general appearance externally, there is a renal capsule, a layer of connective tissue that surrounds it, giving this smooth surface appearance. Let's talk a little more about the location of the kidney. So the term peritoneum is hopefully something that you remember from our earlier discussions back when you would go to BIO201, where we discussed how the different body cavities were layered with connective tissue layers, kind of generating these compartments. Well, peritoneum was the layer lining the abdominal cavity, and since kidney is described as retroperitoneal, that tells us that this structure is now located behind this peritoneal layer. So I hope that that makes sense. One more thing that I'll just point out real quickly, we're not gonna spend a lot of time, is this adrenal gland that we see located on top of the kidneys. Adrenal gland relating and reminding us again of the connection to the endocrine system. If we look at the kidneys, one thing that we'll notice is that students often are surprised how far back towards our back of the body they are located. If we compare the right and left kidney, one thing that we'll notice is that the left kidney is higher. And this is because there is the liver immediately above. So on the right-hand side, liver takes a majority of the space, causing the right kidney to be located lower, so a little more inferior in comparison to the left kidney. In terms of different conditions, we can have issues with the kidneys not being located exactly where we're expecting them to be, or backup of urine, obstructions of various kinds. These are all worth considering. And this is the reason really why we start always with normal anatomy and physiology. So at the time when we get to the pathologies, you're comfortable with what should happen. If we look at the internal anatomy of the kidney, a couple of things that we'll notice there, or a couple of structures that I want to highlight especially. Let's start with the renal cortex. So cortex was the external part, the superior part, similarly as we saw with the brain and few other structures so far. And it is really distinct when you do dissection also in the kidney, this external part. Let's look at then renal medulla. Medulla, of course, the inner part, and one important structure within the medulla that I want you to notice are the renal pyramids. These are cone or triangle shaped structures. Again, pretty often easily distinct in just their tissue characteristics, in just their color. You'll notice those as you are dissecting to the kidney. In terms of other structures worthy of noticing, let's talk a little bit about the renal pelvis. So renal pelvis forms this funnel-shaped structure that collects the resulting urine from the inner parts of the kidney and then directs this urine to a continuation of it known as a ureter. So ureter was responsible for carrying the urine then to the urinary bladder. I wanted to spend a little bit of time to talk about nephrons, because nephrons are really the workhorses of the kidneys. And I believe that both in the class and in our online review sessions, we've now drawn nephrons together quite a few times. So rather than just simply drawing it once again, what I'm going to do, I'm going to take a little bit of a time to just go through the structures, go through the physiology, but this should be revision from what we've covered before. One word of advice, this is just a simplified diagrammatic representation of the nephron. So by no means am I saying that every nephron will have these proportions, it will be running in this direction, and so on and so on. Quite the opposite. What we will end up seeing is that nephrons vary. They can be in any order, any direction. I just simply prefer to use a diagram when we're learning a structure for the first time. But I have had a discussion about this one group of students who were convinced that nephron always starts from the left and ends to the right. And of course, that won't be the case. So let's have a look of the different parts. Here I have a colored diagram that I'm going to use to discuss this. And we're going to start with our afferent arterioles. So afferent arterioles are the tiny blood vessels right after arteries, before capillaries, that bring the blood into the nephron. Then we have our capillary tuft capillary network, which you see as this round structure on this diagram. And we'll talk about that in just a moment. But what you'll notice that leaving from this round structure, we have efferent arteriole. So the smallest vessel type after a capillary network, again, so that we are now talking about these tiny, tiny, tiny blood vessels. One coming in, one leaving. Let's have a look of this capillary network that I talked about. This is going to be called as glomerulus. So that's where the exchange between the blood, between the circulatory system and the nephrons, so if you wish, the functional part of the urinary system that does the most of the work, the workhorse of the kidneys. That's where all that happens. And Bowman's capsule is this kind of a cup-like structure that surrounds the glomerulus. Notice that I've done now a cross-section view of the nephron in this diagram. So it surrounds this capillary network, ready to help with this exchange of materials between the cardiovascular system and the urinary system. Probably another name that you see quite often is glomerular capsule. We're trying to move away in the medical language from people's names to names that actually describe the structure, but you might quite likely see both being used and often being used interchangeably. From the Bowman's capsule or glomerular capsule, which surrounded the glomerulus, this capillary network, we have a tube leaving out of the glomerular capsule. Let's have a look of that tube. This tube will receive the result of this process of filtering blood. And what I do want to highlight at this point is that this process of filtering blood has a specific name. We call it filtration process. So the materials leaving the blood and entering to the nephron, that process is now known as filtration. So our first physiological step of the process here. I was already going to mention about the proximal convoluted tubule. So proximal convoluted tubule, this kind of a thicker, more immediately close after the glomerular capsule, immediately that part of the tubing coming out from there. Then moving onwards, we get to the nephron loop, which is physically a little thinner. This is another important workhorse part of the nephron. So this is where the countercurrent mechanism plays role. So exchange of the materials. Sometimes nephron loop is also known as loop of Henle after the person who first identified it. When we get back from our ascending and descending loop of the nephron loop, we notice that on our first part of this loop, that's more proximal to the glomerulus, we'll notice that material is leaving the nephron loop and returning back to the cardiovascular system. So although we work so hard to move the materials from the blood to the nephron, this is a time where we have a chance to do the opposite movement to ensure that nothing that should not have gone there is going to stay there. So this process of moving materials from the nephron back to the cardiovascular system is going to be called our reabsorption. Well, an opposite process takes place on the more distal part of the loop of Henle, and this is going to be known as our secretion process. During secretion process, material moves now from the blood vessels, so from this arteriole that's running next to the nephron, to the nephron itself and to the distal part of the loop of Henle or nephron loop, if you wish. So this was actually, let me correct it, our third step of the movements of the material. So third physiological stage that we're seeing. And we get to another thicker loop, leaving the loop of Henle. And now we talk about distal convoluted tubule, similar to what we saw earlier on, but this time rather than being proximal, it is going to be distal. And distal convoluted tubule is going to be important for us as we are going to discuss about the what goes on in terms of the presence of antidiuretic hormone. So keep the distal convoluted tubule on your mind just for a second. And the final piece of this tubing, if you wish, is going to be our collecting duct, which receives secretions from multiple nephrons, multiple distal convoluted tubules open to a single collecting duct. So collecting duct is collecting the output, the end result from the nephrons. And this end result is going to be called as our excretion. So whatever is left now to the nephron as the final product after all these steps of moving stuff in and moving stuff out, the final end product is going to be our excretion. So excretions from multiple nephrons are collected to the collecting duct. And here you see all these structures that we discussed labeled in one single diagram. And you'll also see that really the physiology is going to be important for us, because the physiology allows us to get information about how well the kidney is working. So a measure that you'll see on a lot of medical notes, reports, and so on is going to be glomerular filtration rate. And what glomerular filtration rate tells us, it, to summarize, tells that how much blood passes through the glomeruli in each minute. So if you really want to simplify what that tells us, it tells us how well the kidney works. Kidney needs to work reasonably well for doing certain medical treatments, using certain medicines, using, for example, contrast agents in imaging applications, and so on. So the GFR is a value that we would be keeping on eye, that would be important for us. Well, I mentioned earlier on a hormone, ADH, and you might be familiar with ADH from our discussion of our endocrine system. And when we talked about endocrine system and hormone ADH, we defined what it does. So it prevents us from making urine. So it makes us pee less, if you wish. And how this is possible, how this happens, is by ADH making especially the distal convoluted tubule more porous, so materials are able to leave the distal convoluted tubule and move back to the circulatory system. So that's the direct effect of ADH on a nephron level to the physiology. So that's how we end up producing less urine. If we look at the urine itself, we'll notice, of course, that majority of the urine is made of water. We have a small amount of different solutes. These can include many different things. Some we expect and some we keep on eye that we do not want to see. Of course, important ones: urea, uric acid, creatine, that those would be nitrogenous wastes that we are removing from the body with the urine. One thing that we should never find with the urine is blood. So if we're seeing blood in the urine, that is a sign of concern for us. We also are able to tell that there's an infection if we end up seeing white blood cells in the blood. And now this hopefully brings together what we talked about the immune system back in the day. So when we talked about the immune system, one thing we noticed that the white blood cells were present there, but only when white blood cells were needed when there was a pathogen that our immune system activated to deal with, the number of white blood cells increased. So white blood cells are there, but when we find great numbers of white blood cells, that's a sign that the immune system has been activated. There's an active infection that we're dealing with. So now we have talked about the kidneys and we have talked about the nephron. That leaves us with three other structures to talk briefly, because I do want to take a little bit of time to talk about the fluid balance. So let's start our discussion with the ureters. So ureters are these muscular tubes made of largely of smooth muscle lining. The role of the ureters is to carry the urine from our kidneys to the urinary bladder. So you would probably think that gravity is the only force carrying the urine, but if we think back that these are smooth muscle-lined tubes, that helps us to understand that they also do this peristaltic movement, which is this wave-like constriction, which helps to push the urine onwards to one direction and one direction only where we want the urine to go, so towards the bladder. This is why your ureters will still keep working even if you're in zero gravity. So astronauts don't have trouble with getting the urine from the kidneys to the bladder. Again, they are a retroperitoneal organ behind the peritoneum. And one thing to notice is that they enter to the bladder actually at the base of the urinary bladder. So most diagrams like here, we see them showing as it would enter at the very top. That's not the case. They go behind, arch around, and enter at the base of the bladder. So this is what I wanted to show with these arrows. So urinary bladder receives our two ureters at the base, and there is an opening for urethra at the base as well. The ureters more posteriorly, our urethra more anteriorly. If you look at the relationship between these three openings that we see, they form almost like a triangular structure. We actually talk about trigone, this arrangement of these three openings at the base of the urinary bladder. A great directional clue for us when we're analyzing medical pictures. Urinary bladder is muscular walled sac for the temporary storage of the urine. Located right at the pelvic floor. And one thing that I did want to take a moment to talk about is that it is possible that we get infections entering to the urinary bladder. The infections would depend on the urethra, so the infections would typically have to travel up the urethra, and there are differences in the urethra, this tube that carries the urine from the bladder to the external world between males and females. So as you see on the females, the urethra is much shorter than what we find it to be on the males. For this reason, the journey for the bacteria or any other pathogens to travel up the urethra to the urinary bladder is shorter on females, so females are more prone for UTIs, urinary tract infections. The fancy word that I want you to use rather than talking about peeing is when we talk about that process with medical language, we should really talk about micturition. Micturition refers to the expelling the urine, the act of urination. And what I'd like to do next is leave the urinary system and move our attention to the fluid balance, discussing that a little bit as a reminder from the key concepts from this week's chapter. So let's do just that. And one of the reasons why we needed to know about the fluids, where fluids are, what they are supposed to contain, and what they are not supposed to contain, is that knowing the normal allows you to read, interpret results, lab results, when something is not correct. So on this section, we'll talk a little bit about fluids, we'll talk about electrolytes, and we'll talk about the pH. So 3 topics. When we talk about body fluids and we talk about water, one thing that we always remind ourselves is that all living things are made of largely of water, and all living things need water without a few exceptions to stay alive. So the amount of water in a human being, that number varies a little bit depending on what textbook you use, but we're typically looking at numbers between 60 to 70, even up to 75% of your body. What we'll find out is that the amount of water is not the same in all individuals. Infants, newborns have a higher amount of water relative to their body size because they have a low body fat, low bone mass, and so on. If we compare that to the older people, older you get, less water content there is in your body. We can also talk about males and females and see that there is a slightest difference between the two. Males having a little bit more water in their body than their females. This difference really depends mainly on the females having a higher fat content, less skeletal muscle mass, and so on. But really, what we notice is that majority, more than half of our body content is water. So water is clearly important for us. Where that water is, where it goes, and what happens to it. Well, if we talk where that water is, one thing that we'll notice is about 2/3 of all the water in our body is within the cells. So we call that water content intracellular water. The remaining about 1/3 of the water is extracellular, so outside the cells. And this is mainly the interstitial fluid proper, so the fluid between the cells, but also blood, plasma, and our lymphatic content. This list, I believe, should be largely a revision from what you remember from when we've discussed about what makes water so essential for life in your introductory classes to the biology. But let's take a moment just to remind ourselves of that. So water is an excellent transport media, and we'll talk about all the things that can get dissolved into water and how that is important at the end of this list. But at this point, I do want you to notice that most things that we use to transport stuff in living organisms, such as, for example, blood in animals or water just transporting nutrients in plants, do use liquids that are mainly made of water. Of course, we talked earlier on about the important role of blood in transporting so many things in our body, including respiratory gases, nutrients, waste products, hormones, and so on and so on. But notice it's not just the blood, it is really water in the plants as well. That is the main transport media. The concept of cohesion and adhesion is something that I assume that you have heard before. So cohesion refers to the water molecules' affinity to each other. So water molecules tend to stick together. When we talk about adhesion, water molecules also tend to stick to polar surfaces, so anything with a charge. So these two forces are very important in how water can help us, how water behaves, why water is so important. These two forces also contribute to the surface tension on the surface of water bodies. For example, different bugs, we see them walking on water thanks to this high surface tension due to the cohesion of the water molecules. One of the big things that we notice is that water is excellent at binding heat, binding warmth, and then releasing it slowly over time. This is one of the balancing reasons why the temperatures, the climate is kind of consistent on the beach areas. So during the hot day, big bodies of water tend to bind a little bit of that heat, and during the colder nights, they release that heat. So the heat fluctuations are lesser. That's why many people prefer to live on the coastal areas. That's why we see these sharp changes between super hot roasting summer days and freezing cold nights here in Arizona, where we have limited amounts of humidity and bodies of water. Water, of course, could take three phases. There is a fourth phase, not so much relating to the water, but certain other substances. But we're going to stick with the liquid, gaseous, and solid phases of water. And like I mentioned earlier on, water is an excellent solvent. In fact, we describe water often as the universal solvent. So many things can dissolve into water or can be carried by water. So let's have a look at those solutes a little bit. There is a bunch of stuff carried by water that does not really dissolve into it. Important ones for us in medical field, glucose, lipids, urea, creatine, and so on. We could continue that list. They still nonetheless get carried into water. So water works as a transport media for these. The ones that we are going to be most interested are the ones that actually dissolve to the water. And the reason why we're going to be interested about these is that as they dissolve to the water, there's going to be change in the charge of these substances. And that's important for us as we move forward on our discussion. We can, of course, find various different dissolved charged particles at different parts. The two that I'm noticing on this list are sodium and potassium, whether they're within the blood or intracellular or extracellular. That is important. You remember, for example, our sodium-potassium pumps that systematically pumped sodium and potassium to different directions to generate this charge difference inside the nerve cells, about minus 70 millivolts as a charge for the inside of the nerves. So really, these charges are going to be important for us. There are a few other ones that are worth looking at as we're talking in more detail. If nothing else, the one thing that I would like you to take away from the portion where we looked at the fluid balance and electrolytes is that water and these charged particles, these electrolytes, they are interconnected. So as one moves to one direction, the other one typically tends to follow. And this will be very important for us, as you'll see from the couple of examples that I have listed. But before we go to those examples, I do want to talk in general about the general water intake. So in a day, typical average person should be taking in about 2.5 liters of water. That should be equal to the amount of water that the typical individual puts out as well. So as long as intake and output are equal, we should be in a good place. The main driving force for intake of water is going to be thirst. So we drink most of the water that we get into the body. Some we also get through the moist food products that we eat. You might wonder, what is this metabolism that results in some water? So remember the reactions where we break a larger molecule into smaller components, and as a result, we release energy. And another thing that we released as a byproduct of this process was always water molecules. So that's how all metabolic reactions in the body contribute. If we are producing water as a byproduct of those reactions, they contribute to the total water intake, or more of the amount of available water in the body. The ways we get rid of water, well, that's going to be mainly through urination. So the process of expelling urine micturition. We do lose some through evaporation, sweating, and breathing through processes, and smallest amount through feces. So you might be surprised how little water we lose through feces. And we'll discuss how that digestive system is as specialized, how the large intestines especially are responsible for the recovery of the water from the feces before they are expelled from the body, as we move on to the further chapters on this course. We should talk briefly about how do we regulate the water balance in the body. What is the maintenance mechanism? And different textbooks will give you different kinds of diagrams, and these diagrams can be sometimes more complex, sometimes a little less complex. They can include a lot of anatomy, physiological structures that you've seen. But if you think back to the very beginning of our course, I said that keep it simple. Learn this one concept and it will help you greatly. And this one concept that I encouraged you to learn was, of course, the concept of homeostasis. So homeostasis referred to the maintenance of a consistent internal environment in the body. So we want to keep the internal environment of the body consistent, and that was done through a four-step process. We had a stimulus, which was the chains, that was detected. Oh, I didn't mean to do that. That was detected by a sensor. And let me just get my marker out. So some sort of a change that's detected by a sensor, which passes the message to the control center, usually our brain. We have some sort of an ideal range, normal range of values that we would expect the stimulus to be. So we compare the data from sensor against that. And if necessary, we recruit the effector. Typically, we try to stop the stimulus from happening. So most homeostatic mechanisms are relying on negative feedback. Rarely we see positive feedback, but we discussed about a few of those examples. Let's have a look at what happens when there's too much water in your body. Well, this gets detected by osmoreceptors, which are located at the hypothalamus of your brain. This gets compared to the normal, against the normal amount of water that we would expect to see, for example, in the blood. And if necessary, the pituitary gland will start secreting less ADH. Remember, ADH was anti-diuretic hormone. So as a result, more water now goes out from the body, more water is excreted, so we end up peeing more. More pee is being made in response so that we can get rid of some of this extra water. Opposite chain of events, if we have too little water, again detected by the osmoreceptors at the hypothalamus, compared against the normal range within this comparison process happens within the brain, processing this information. If necessary, now we recruit our pituitary to produce more ADH. So more water is taken back at the distal convoluted tubule, and as a result, less pee is being produced. So you can really see how the hormone system and the urinary system work together for the physiology of maintenance of water balance. I hope that that kind of restored your faith in that all of these things that we talk about are actually connected to each other. Let's take a moment to talk about tonicity. And tonicity is something that I think that you might have discussed earlier on in your previous classes, but it is a key concept, especially when we talk about the water balance in the body and when we talk about the medical aspects. We do need to be very clear of this concept. So I want to take a little bit of time to discuss about it, and it is also included in your exam. So a little heads up there. And let's start with condition that's known as being hypotonic. But before we jump into the hypotonic condition, I just want to remind you that this movement of materials is due to diffusion. So this is a passive process where materials from areas of high concentration move to areas of low concentration. So if we add a food coloring, for example, to this glass of water, there's going to be high concentration of food coloring in the area where we add it. But slowly, the high concentration will decrease and the concentration will even out throughout all of the class as the movement of materials is just spontaneously due to the diffusion from areas of high to areas of low. So this diffusion passive transportation process is going to be the key driving force for what we'll see for the movement of waters in these different tonicity conditions. And I'll add to the end of this video a link to another classroom video that I've done actually in one of my classrooms. I think it's for introductory biology course for allied healthcare practitioners about the tonicity. So hopefully that will help. And let's look at the hypotonic condition. In hypotonic condition, now the inside of the cell is going to be more salty, or there's going to be more of these electrolytes than on the outside of the cell. Remember, body always try to go from areas of high concentration to the areas of low concentration. In this case, there's going to be more water outside than there is inside the cell. So water will flow into the cell. As a result, the cell will start swelling and growing bigger as more water flows in. In an extreme case, it could be possible that the cell ends up bursting with the excess water flowing in there more than its walls can hold it. Well, how do you remember hypotonic cells? Always, when possible, make mnemonic rules, make things that help you to remember complex concepts in a simple way. So when I think of hypotonic cells, I think of these cells that are big, swollen, kind of chunky looking cells. And I think of an animal, I think of a hippo. So hippo referring to the hypotonic cells. Hippos are these big, swollen looking animals. So hopefully that will help you to remember that. Let's next have a look of isotonic cells. In isotonic environment, there is a balance between salts both inside and outside the cell. So some water will be entering the cell, but equally some amount of water will be leaving the cell. So the overall gain and loss are in the balance. The cell remains as the same size. It doesn't shrink, it doesn't swell, it just remains as it is. So the amount of water coming in, amount of water leaving out is in perfect balance. So if I think of isotonic cells, how can I remember that? Well, since these cells are perfect as they are, they don't need to change, they're not gaining, they're not losing. I think that these cells tell me that iso-perfect. So isotonic cell is a perfect cell, iso-perfect. I hope that that helps you to remember that. If not, come up with your own mnemonic by all means. The final type of tonicity that I wanted to discuss is going to be our hypertonic cells. In hypertonic cells, the outside of the cell is actually more salty than the inside of the cell. Remember, we always try to balance off the differences, moving from areas of high concentration to low concentration. In this case, water being in higher concentration inside the cell than in the outside. So water flows out from the cell. And as a result, the cell may end up shrinking or eventually, in the worst case, totally drying up. So how to remember this hypertonic cell referring to cells shrinking and drying up? Well, I think of what sort of a person is a person who's hyper. Hyper people have a lot of energy. People who have a lot of energy, what could they do? Well, they run around a lot. What happens if you run around a lot? You become very skinny. So hypertonic cells, in my mind, are skinny cells. They are shrinking. So I hope that helps you to remember those. One thing that you'll notice, for example, if you exercise a lot and you sweat a lot, you lose a lot of water. With that water, you will also lose a lot of electrolytes. After exercising, you get this craving for salty food. That's because even though you've been replenishing normally the lost water by drinking more water, you might not have been replenishing the electrolytes that you lost with the sweat. So that's just your body's innate response to kind of promote you, make you wanna get back those electrolytes. Alternatively, it is possible to die out of drinking too much water. In fact, back in, I want to say, early 2000s, mid-2000s, there was a competition in one of the radio stations where I think it was called Hold Your Wii for a Wii, the game console Wii, that was just rolled out. And the concept was that the participants were asked to drink a lot of water in a short period of time and not go to bathroom. Well, I do wish that everyone had taken their basic classes in basic biology. We would know that this would be dangerous because now we would have a risk of individual becoming hypotonic, water flowing into the cell, and that would be bad. This is normally not a risk for an average individual, as long as you don't go crazy with the amount of water that you drink in a very short amount of time, and as long as you don't prevent yourself from going to the bathroom. Consciously holding the urine is stopping your body from doing these regulatory mechanisms that we saw earlier on for getting rid of additional water when it's too high concentration. So really, that was just a stupid competition, not thought through, and actually resulted in one of the participants passing away as a result of that, dying from taking part on this competition. So be careful. Learn your basic bio, I guess, is my moral of the story on this one. Like I said, sometimes you exercise a lot, and as a result, you might end up sweating and trying to replenish that lost fluid by drinking a lot of water. As a result, your body fluids might become too dilute. Your brain actually tells you to urinate to get rid of this diluted state, and you actually become more dehydrated, although you just drank some water. So it can be kind of a vicious cycle. So what athletes who really exercise so much that to lose some of the heat, they have to sweat, and as a result, they lose fluids. What they do, they want to make sure that they drink drinks that contain electrolytes. So these electrolytes must be replenished as you sweat them out. Just regular water might not be ideal. Sodium chloride and chloride are some of the important ones. So I have here a list of different recovery drinks and how much of different components they contain. You really notice that orange juice, soft drinks are rather poor for this purpose of replenishing electrolytes. You really want to go for the drinks that are aimed for these recovery drinks, such as Gatorade, Powerade, and so on. So they serve a purpose, but I would really encourage you, these are for the people who exercise to the amount that this becomes an issue. A regular person on day-to-day life likely is not likely to sweat so much that replenishing electrolytes is a huge challenge for us. To wrap up this session, I want to mention a couple of words about the pH scale. We're not going to go into great detail, but I do want to remind you of this concept. So a pH scale measured the concentration of hydrogen ions. And remember that this was a logarithmic scale. So as we move one number unit up, it's actually tenfold that the concentration has increased. pH scale with the neutral being at 7, and we are alkaline. Acid, if we have higher than 7, if we are below 7, we are then basic. What I want to say or highlight here is that notice how the normal pH of the body is maintained within this very narrow range between 7.35 and 7.45. So 0.1 is the difference, and this is really a remarkable accomplishment. If we try to teeter mix our own solutions in a lab setting from acidic and basic substances and try to get into this pH of a healthy body pH. But one thing that we'll notice is that it is very hard to do. So your body must have some special adaptations to be able to maintain the pH within such a narrow range of, or such a narrow window, such a narrow range that is optimal for enzymes, proteins, many chemical reactions, and so on to work in the body in an optimal state. So let's have a look of how that would happen. I just want to mention first, and we'll come back to this, that if we are going off from this normal range, we can end up with alkalosis or acidosis. This would be a problem. Eventually, worst case scenario can lead to death. So there's 3 methods how we are maintaining the body pH so nicely controlled. And one of these is a rapid response, whereas the 2 others are slower to respond. The 2 others are though more efficient in terms of their potency as responses. So we need all 3, and it is balancing act of all these 3 to maintain the body pH where it needs to be. So let's have a look of these together. And we're going to start by talking about buffers. So buffers are these chemicals that bind to acids or bases, bringing the pH to closer to neutral. We talked about these buffers in introductory biology classes. These are the first line of defense and very quick way of responding to changes of the pH in your body. At least in my classes, we try out various different substances and try to figure out what would be good buffers and what are not so good buffers. So if you haven't had a chance to do that, I really do encourage you, and I might be able to link a link to that lab to the end of this video as well, provided that I find it. So I'll do that for you. In terms of the 2 other methods for regulating the pH of the body, well, first of all, respiration. So we can try to deal with the excess buildup, excess concentration of the hydrogen by changing your rate or depth of respiration. This is slower, takes several minutes to act, so not as rapid as buffers as a respiration. And the slowest way to deal with this excess hydrogen is by urinating them out. This is really slow because it can take from hours to even days to act, but you do get rid of a huge number of hydrogen at once by urinating those out. So these are the 3 mechanisms, and it is the combination of these that really allows us to maintain the hydrogen ion concentration in the body within this normal range so well. I promised to mention a little bit about acidosis and alkalosis. I've listed some signs and symptoms, consequences as well on these on this slide here with the two sides showing of different conditions. I guess that my biggest take-home message from this would be that worst case scenario, either one can lead to death in an extreme case. So we don't want to go off too far. This, I believe, is the very last slide in this set of slides that I've prepared for today. I do want to remind you that often when you go to doctors, the medical facility, one of the first things that we do put a fluid drip for you to your vein. So we administer fluids. This is often because patients are dehydrated. So this method allows to respond to that. It also helps to maintain the blood volume up. Of course, it's not ideal in terms of the concentration of the blood, but at the short term, it's a great way to bring up your blood volume. Of course, this fluid that we are getting into the patient cannot be just any old fluid. It cannot be just regular water. It needs to have a right balance of electrolytes in it. So that we don't end up with hypo or hypertonic environment. So that's why it was so important for me when we discussed about the tonicity that you are comfortable with these three concepts. They are really crucial. You could do a lot of damage by administering intravenous fluids that would not be in correct balance of electrolytes. And that really concludes everything that I had planned for this week's review session. I will be repeating this review session in the evening as well. If no one shows up, I might just simply upload this video again. But if someone shows up, I'll be here and I'll be happy to discuss the concepts from last week and this week again, just to bring us all up to the speed. So that you are all ready for tomorrow's laboratory class, where we'll look at the fluid balances by doing some application of that in a little bit more of a clinical approach of looking the information there. So I hope that you have found it useful, and I thank you for joining me, and I really hope that you have a very good rest of the day. Most of you, I hopefully see tomorrow in the class in person. Until then, thank you and bye.